Now we are going to learn about protein synthesis, a process that's going to be taking place at the ribosomes in the cytoplasm when proteins are going to be needed. We are also going to be learning about transcription, which is going to involve a second nucleic acid called RNA. Now, we learned that DNA is going to be made up of nucleotides, 3 units, a phosphate group, a 5-carbon sugar that in DNA is going to be deoxyribose, and a nitrogen base. RNA is also going to be made of nucleotides. The difference being now that in RNA, the sugar is going to be ribose. The phosphate is going to be the same as in DNA. And the nitrogen bases, there will be one difference. The nitrogen bases that we find in DNA have been identified as thymine, adenine, cytosine, and guanine, and they are going to be complementary to each other with A bonding with T, C bonding with G. In RNA, the bases of adenine, cytosine, and guanine are going to be the same as they were in DNA, but now we have uracil instead of thymine. So the complementary base pairing for RNA is going to be A with U and C with G. Another difference is that the DNA, as we mentioned, is a double helix containing 2 polynucleotide chains. The RNA is going to only contain 1 polynucleotide chain, and that's going to be specifically in messenger RNA. Another difference is where they're going to be found. DNA is going to be found only in the nucleus. RNA will be made in the nucleus, but then move out to the cytoplasm. In addition, there will be 3 types of RNA found in the cell. The first one that we've been describing is called mRNA, or messenger RNA. The job of messenger RNA is going to be to copy DNA's code during transcription and take that information out to the ribosomes where the proteins will be manufactured. The DNA, the sequence of nitrogen bases in the DNA, are a recipe, a code for the production of proteins. But since the DNA does not leave the nucleus, it'll make messenger RNA. So messenger RNA will take that code out to the ribosomes for the proteins to be manufactured. A word that you need to remember to associate with messenger RNA is codon. Codon is going to be 3 nitrogen bases on messenger RNA, and those nitrogen bases are going to be coding for an amino acid. The other type of RNA is going to be ribosomal RNA, represented by rRNA. Ribosomal RNA is going to make up the ribosome and assist in the translation process. The third type of RNA is called transfer RNA, and transfer RNA's job is going to be to pick up a specific amino acid and bring it over to the ribosomes when the proteins are being manufactured. Amino acids are the building blocks of proteins, and they will be lined up in a proper sequence as dictated by the codons in the messenger RNA. Anticodon is the word that you need to associate with transfer RNA. Each transfer RNA will have 3 specific nitrogen bases at one end that we call an anticodon. And at the other end, it will have a specific amino acid. Anticodons are going to bond to codons, complementary base pairing, where now, because we're talking RNA, A is going to bond with U, C is going to bond with G, in order to line up the proper sequence of amino acids to make the protein that is, whose recipe is found in the particular gene that's being read at the time. So to summarize the three types of RNA, messenger RNA, or mRNA, is going to be complementary to DNA. So the complementary base pairing is going to be C with G, A with U. This is going to be made in the nucleus during the process of transcription, and then it'll travel from the nucleus to the ribosomes in order for the protein to be manufactured. That's going to be its job, direct the synthesis of protein, since DNA does not leave the nucleus. Ribosomal RNA, also called rRNA, is going to be found at the ribosomes and will assist in the translation process. And then transfer RNA, or tRNA, its job is going to be to bring an amino acid, a specific amino acid, to the messenger RNA during the synthesis of proteins. When a protein is needed by the cell, the process of transcription will take place, whereby messenger RNA is going to be made from DNA. The process is going to involve unwinding, opening up only a section of the DNA, not the whole DNA as we saw in replication. That section of the DNA that contains the sequence of genes that code for a particular protein is going to be called a sequence of nitrogen bases that code for a particular protein is going to be called a gene. So in this diagram, this area here would be the gene, this whole area. And that's the only section that's going to open up. The enzyme that's going to be helping during this process is going to be called RNA polymerase. It's going to help bring free nucleotides that exist in the cytoplasm coming from the food that we have consumed. And they now will be bonding with their complementary bases, as we see here in this picture. The R in A is represented with the red being the ribose and phosphates making up the backbone of the molecule, and then the free nucleotides are joining with their complementary bases. And remember, now in RNA, we are not going to have thymine, we're going to have uracil, and uracil will bond with adenine. So the process will continue until all of the DNA triplets have their messenger RNA complementary bases bonded to them, and the molecule of RNA is complete. The RNA polymerase will join the sugars and phosphates, and once the molecule is complete, it will move out of the nucleus into the cytoplasm. During the transcription process, RNA must first be modified before it leaves the nucleus. That is going to involve eliminating segments called introns that do not carry necessary information for the protein formation. The introns are going to be removed, and the exons are going to be spliced together, and that's what's going to be leaving the nucleus and heading to the ribosomes for the actual protein synthesis, a modified version containing just exons. So once the messenger RNA is formed in the nucleus during transcription, it will move out of the nucleus and land on a ribosome. There it will be read and decoded and the proper sequence of amino acids brought into position during the process of protein synthesis, also called translation. So we start with the messenger RNA, and it's going to be read in groups of 3 nitrogen bases called codons. So here is a codon on the messenger RNA, UUU. In the cytoplasm, there will be transfer RNAs. Each transfer RNA will have an amino acid at one end, and at the other end, it will have 3 nitrogen bases called anticodons. So here would be an anticodon on a tRNA. 2 tRNAs at a time will come in, bond with their complementary bases, complementary anticodons on tRNA with complementary codons on mRNA, and thereby bring appropriate amino acids into position. Those amino acids now will be bonded. A special bond called a peptide bond will form between the amino acids, giving rise to the protein that will be formed. The sequence of those amino acids will determine the protein, and the sequence of the amino acids will be determined by the sequence of bases on the mRNA. And this sequence of bases was determined by the sequence of bases in the DNA that transcribed this messenger RNA. This table will show you the messenger RNA codons and their corresponding amino acids that they will be coding for. We are going to read the table starting on the left side with the first nitrogen base in the codon, then along the top for the second nitrogen base, and then along the right side for the third nitrogen base. Now, RNA has 4 nitrogen bases: adenine, uracil, cytosine, and thymine. 3 are read at one time in a codon. That makes for 64 possible codons, 4 nitrogen bases to the 3rd power. And that's what's showing here on this table. As you can see, some codons will code for some amino acids, will be coded by more than one codon. Now, I have something here at the bottom of this table to help you understand how to read this. If we start in the DNA with the triplets that are shown here, for example, if the DNA said TAC, that would be transcribed to mRNA with a codon AUG. Now, if you look at this table, A being the first nitrogen base in this codon, U is the second. We look down and across in this table, and the third one you're going to read over here, G, and that gives us methionine. Methionine is always the first amino acid. It's called the initiator or start codon. That indicates the beginning of a new gene. Now, what transfer RNA would be bringing methionine? What would be complementary to AUG? It would be UAC. And that's the tRNA that would bring methionine into the position where the AUG was found on the mRNA. If the DNA said TGG, what would be transcribed from that? T would go with A for the RNA, G with C, G with C, so you would have ACC. Looking again at the table, A, C would bring us into this table here. C, ACC, the codon for threonine. And what anticodon on tRNA would bring threonine in? What's complementary to ACC would be UGG. Another example. If the DNA said CGT, what would be transcribed from there? GCA. Looking at the table, G, C, A would bring in alanine. And the tRNA that would carry alanine would be complementary to the codon. That would be C, G, U. The last one that I have for you here is if the DNA said ATT, that would be transcribed with UAA. And if you look at the table, UAA would be a stop codon. So the tRNA that would bring the stop codon into position would be AUU. Every molecule, that is every protein that is produced, the last codon is going to code not for an amino acid, but a stop codon. And that indicates that the protein is complete. It's ready to break off and go do its job in the body. So what we had going on here from DNA to mRNA, this was transcription. In order to take DNA's code and make mRNA from it. Then the mRNA codons are being read and translated at the ribosomes during the process of protein synthesis, also called translation. And your end product is the protein that's being formed according to the sequence of bases on your mRNA molecule. Protein synthesis requires 3 steps. Initiation, when the mRNA binds to the smaller unit of 2 ribosomal units. Every gene will start with the initiator codon AUG, which codes for the amino acid methionine. Elongation, where the polypeptide chain lengthens 1 amino acid at a time. 2 tRNAs can be at the ribosomes at one time. As their anticodons pair with their complementary codons on mRNA, the amino acids they carry are in close proximity and they form a peptide bond. This process continues until all codons are translated and the proper sequence of amino acids are lined up to join and form the protein. And then the last stage is termination, where the final stop codon does not code for an amino acid. Instead, it indicates the polypeptide or protein is complete. This picture from your textbook serves as a good review of the whole process. So starting with DNA in the nucleus serves as the template to make the mRNA, which will be processed and modified before it leaves the nucleus. When it leaves the nucleus, it will land on the ribosomes, where tRNAs with anticodons at one end and amino acids at the other end will come and bond. Complementary base pairing will occur between anticodons and codons to bring the proper sequence of amino acids into position, and then they will be bonding to each other, forming a peptide chain. 50 or more amino acids would make a protein, and upon completion, once the stop codon is read, the protein will depart and go and have its function in the cell, and the tRNA will go and pick up another amino acid and wait until it's needed in the process again.